Who are we missing? Jennifer, Sam, Les, Hannah, and Jenna. Are you going to do?
Yeah, I'm gonna go over the quiz first.
Okay. I have stuff, intro stuff.
Okay. Actually, I did too, so I'll let you do yours.
Okay.
Cool, Dad, I will. Okay, welcome back. Hopefully you are all refreshed. I know lab quiz seems like probably just forever ago, but I want to go over the lab quiz with you. This is pretty much the same lab quiz that is given every year. Class mean for the lab quiz this time was 26.6 out of 35, which is 76%. And that compares very favorably to all of these past years that I just dug out right here. So there's a little variation from semester to semester in the lab quiz. You can see that for lab sections, there's not much variation as well. Standard deviation is pretty low. And when you look at t-value comparisons, remember 2 is kind of the magic number. If your t-value is somewhere around 2 or above, then that might mean a significant difference. So there's really no significant difference in performance in any of these lab sections. Let's run through the answers to the lab quiz real quickly here. Maybe. Yeah. Before we do that, one very important reminder. We're doing the P-Glow lab this week, which Dr. Morton is going to introduce in just a minute. But it's really important that you read that lab prior to coming to lab, okay? Remember, we're not doing our presentations this week. We gave you a break and allowed you this week to work on presentations. I would bring your presentation material to lab this week because this P-Glow lab really probably won't take the full 2 hours, will it? And so that would be some time you would have with your lab partner or partners to work on your presentation readiness. Okay, the first 2 questions on the lab quiz asked you to identify some organisms that you had seen in class. And these are the answers to the first lab question, number 1. And then these are the answers to the second lab question, number 2. So that was just really a matter of identification. Okay, the third question was a multiple choice question, and it asked which of the following is not a reactant or substrate in the peroxidase enzyme reaction. And the correct answer is tetraglycol. Tetraglycol is that oxidized product of the reaction, the stuff that turns brown. And so that was not a reactant, that was a product. This gives you your baseline peroxidase enzyme assay, with the contents of tube number 1, tube number 2, and tube number 3. So if we did an experiment to double the amount of enzyme is the turnip extract, which of those tubes would we have to change? We would have to change the ones that have enzyme in them. So that would involve changing both tube number 1 and tube number 3. Okay? Remember, this tube has a total volume of 10 mils. These tubes have volumes of 5 milliliters. So we've got to keep those volumes the same. So we would change tube 1 and 3. Tube 1 would have 0.1 glycol, 2 mils of enzyme and 7.9 of water. And then tube 3 would have 2 mils of enzyme and 3 mils of water. Why is it important for a cell to be able to produce a peroxidase enzyme? Because during normal cellular metabolism, organisms produce hydrogen peroxide, which is a toxin. And so this peroxidase enzyme is going to convert that toxic product into harmless water and oxygen. Okay, we had our table of foods experiment, which is just like the one that you did in lab at the beginning of the semester. Benedict's test, red color. Biuret test, purple-violet color. Iodine tube, yellow-brown. Sudan 3, no colored spots. So which of these were a positive result? That's a positive result. That's positive. These two are negative. So based upon these results, can you tell what the food is? Yes, you can. The only food that fits those criteria is powdered milk. And one of the notes that you have to be aware of is that in that table, numbers in parentheses are considered to be negative values. So even, for example, maple or powdered or potato starch has 8% protein. That is considered to be a negative value. So the only ones that give you positive results for protein and reducing sugar are the powdered skim milk. We had our dialysis bag experiment, and instead of reading all the text, here's basically what we had. Before the experiment, we had a bag with starch and glucose inside the bag, iodine out here. And then after, what did we have? We had a clear solution in the black bag and a blue solution out here. And then we did Benedict's test on the contents of the bag and the contents of this liquid out here. Benedict's test was positive in both cases. The glucose started here. Some of it ended up here, so that membrane is permeable to that glucose. But the iodine did not get into the bag. Otherwise, the contents of this would have turned blue. So it's impermeable to iodine, but it did turn blue out here. And so this membrane is permeable to starch. So the correct conclusion is permeable to starch and glucose, impermeable to iodine. Hypothetical yeast experiment. These are not the same results that we got in lab, but based upon those results, you were asked to determine whether the cells in flask B were permeable to sodium carbonate. Explain why. And then do the results suggest that the neutral red entered the cell by active transport? Again, explain why. So we really didn't need these results over here. It only asked about flask B, not flask A. So what did we do? We mixed the neutral red and the sodium carbonate together, and we got that orange-yellow color. That's what we would expect. That's what you got. Okay? That's the color that neutral red is in a basic environment. Then we added the yeast. The color was still orange-yellow. That's not what you got. You got the red color. Why? Because the neutral red was entering the cells, which it still is here. But in our experiment in lab, it turned red because the sodium carbonate wasn't getting inside and neutralizing that acidic cytoplasm. In this situation, the sodium carbonate was entering, though, and consequently it neutralized that acidic cytoplasm. Okay? We filtered. The cells were orange-yellow. We acidified them, and now they turned red. Okay? So there is neutral red in those cells, but it wasn't until we acidified them that they turned red, because there's sodium carbonate in here. Okay? We took some of the solution, and this is orange-yellow, and we acidified it, and it turned red as well. So there's neutral red at the end of the experiment in the solution and in the cells. Is that active transport? No. Active transport happened in our experiment because all the cells were red and there was no color left in the surrounding solution. That was active transport. So this would suggest diffusion. Okay, so they were permeable to sodium carbonate because those cells were orange, not red. And then after acidification, the cells in the solution were both red. And that suggests diffusion. The experimental procedure that we used in lab to investigate the question, how does temperature affect beet membranes? These are some of the elements that you should have talked about. You should have talked about taking out beet cores, exposing them to different temperatures, putting them in the refrigerator, water, etc. Soaking them for 30 minutes to allow those pigments to leak out of damaged membranes, and then taking absorbance readings. Procedure for blanking the spectrophotometer, and then why is it necessary to do that? Well, for blanking the spectrophotometer, we need to set the wavelength to the proper wavelength. We need to take a tube of solvent or a control, put that in the cuvette, put that in the spectrophotometer, and then set the spectrophotometer to 0 absorbance. Remember, we took absorbance readings, not percent transmissions readings. Why do we have to do this? Because those solvents or controls will have substances in them that will absorb light. And so we want to cancel out any absorption that's due to the solvent or the controlled materials. It's almost like tearing a balance, okay? When you tear a balance, if you put a weighing boat on there and then hit tear, it subtracts out the weight of the weighing boat and now reads 0. That's effectively what you're doing to the spectrophotometer. Now when you get a reading, it is all due to the products that are formed in our enzymatic reaction. Okay, we've got some data for you with CO2 production at pH 3, 5, and 7. Here was our data. So things that you needed to know were N values. We're comparing two means, so degrees of freedom is N minus 1 plus N minus 1, so 18 degrees of freedom. The 95% confidence level from the T-table for 18 degrees of freedom was 2.101, and the 99% confidence level was 2.878. So, which differences are significant? We've got 3 versus 5, 2.248. That is significant at 95%, but not 99%. We've got over here 5 versus 7. That is not significant at either 95 or 99. And then we've got this value right here of 3 versus 7, 2.848. That's significant at 95%, but not quite at 99% confidence. So your correct answer was 3 versus 5 significant at 95%, 3 versus 7 95%, 5 versus 7 not significant at either level. And then finally, the question that was from your lab handout about the RFs of 1.2. How would you comment, explain? You should comment that that's an impossible value because RF is determined by the distance of the substance traveled divided by the solvent. So if you have an RF of greater than 1, the amino acid would move faster than the solvent, but you can't do that. If you have any